first of all, in the bottom part of this slide, you can see my address and my phone number. It's there because if you are interested in discussing any of these of the things we are going to talk about today, you are welcome to contact me in any way you prefer. The other thing is, and this is not a routine way of saying, if you do not understand anything or something, sorry, not anything, or if you have a curiosity, a question, or a comment on what I'm saying, please do interrupt me. Questions are welcome and a sign or an indication of interest. So I'm pleased to receive and answer questions. So our theme today is the control of the cell cycle in terminally differentiated cells. What you see here is a schematic that in its infinite variations must be, I'm sure, is familiar to you. And that's the classic view of differentiation, which entails a cascade of events, a cascade of successive states from the, let me switch to laser pointer, if I can. Strangely, it doesn't work. Never mind. So, in the classic view, differentiation is unidirectional. We start from any sort of relatively or very undifferentiated state and go down through a number of different states that are progressively more and more differentiated until some lineages, if not all of them, reach a state called terminally differentiated. Terminally differentiated cells are those cells, we'll see that have lost the ability to proliferate in the process of differentiating. Extreme examples of terminal differentiation are, for example, erythrocytes or skin cells that in mammals lose their nuclei in the process of differentiation, and so they are obviously unable to go back to a less differentiated state. But these are exceptions. The classic view that differentiation is unidirectional, that cannot go back, that cannot be reverted, has been challenged by the discoveries we have made in the last several tens of years. So it is no longer true that differentiation cannot be reverted, both in nature and in the laboratory. And I'll try to show you how that can be done in the lab to apply what we learn in the process to regenerative medicine. So our bodies contain a large number of terminally differentiated cells. For example, neurons, cardiomyocytes, inner ear cells, sensory cells that are responsible for our sense of hearing, myofibers, adipocytes, white blood cells, and so on. A large number of cell types that are terminally differentiated. Now that must be qualified. These are classically considered terminally differentiated, but as we will see in a few minutes, they are not necessarily postmitotic, as we say. Postmitotic means just what I've said, that cells in the postmitotic state are unable to proliferate anymore, have lost permanently the ability to proliferate. So if we're going to talk about terminal differentiation, we should try to define it. And again, the classical definition of terminal differentiation rests on two pillars. Terminally differentiated cells are characterized by specialized functions and permanent proliferation at rest, classically termed irreversible proliferation at rest. Now, I should point out that both branches, both pillars on which this definition rests, are in fact quite questionable. Let's talk about the first one, specialized functions. Well, all cells are specialized in some sense. Compared to a totipotent zygote, all cells have some degree of differentiation, and we don't really know nor understand what degree of differentiation, if any, requires the postmitotic state, requires exiting permanently from the cell cycle. So, calling a cell specialized is rather subjective, and there are highly specialized cells, such as hepatocytes, for example, that are not terminally differentiated. So this is one problem. Now, what about permanent proliferation at rest? Permanent proliferation at rest basically is a negative definition. It's based on negative evidence, which in science is always more difficult to prove than positive evidence. If you see something, you know it's there, you know it exists. If you don't see something, it doesn't necessarily imply that that something does not exist. It may just be that you haven't looked hard enough for it. So, as you can see, both pillars rest on soft ground, and the definition of terminal differentiation is not really solid. Now, I think you all agree with the subjectivity of the term specialized functions. But what about permanent proliferation at rest? You might think that my objection to a negative definition is rather academic. Yes, in theory, cells that you cannot see proliferating either in vivo or in vitro might conceivably proliferate in some rare circumstances, but that's, unlikely. Well, it is not. There are cells that we have classically considered terminally differentiated that are not, or are not totally so. For example, this is a paradigmatic example. Cardiomyocytes have long been regarded as completely postmitotic, terminally differentiated, but about 30 years ago, a cardiologist, Piero Anversa, who has an Italian name but is American, showed that if you look hard enough, you can find proliferating cardiomyocytes in the context of intact hearts, and even more in the context of failing hearts, hearts that no longer are strong enough to pump the required amount of blood. So what you see here in these immunofluorescence pictures are mitotic cardiomyocytes. Cardiomyocytes are distinguished in these slides from the other cells that make up the cardiac tissue by this red stain, which is an immunofluorescent stain for troponin T, a protein only expressed by cardiomyocytes. So in the black spaces here are cells that are not cardiomyocytes. And I think it is clear enough that you can see mitotic figures within red cells, within cardiomyocytes. Now, why has this been not noticed for a long time? Why scientists have considered cardiomyocytes as terminally differentiated cells? Well, simply because they divide very, very infrequently. So infrequently that by sophisticated methods has been calculated that during our whole lifetime, only about half of the cardiomyocytes ever get replaced by new ones through division of the pre-existing ones. So only half of our hearts at the end of our lives will have their cells replaced. So this fits with the objection I've raised a minute ago. That is, until people looked deep enough and looked patiently enough for very rare events, they thought cardiomyocytes could not divide, but in fact they can, even though infrequently and slowly. But this is not the only example. Here, for example, I'm showing neurons, prototypical examples of what we regard as terminally differentiated cells. But in this case, you can see neurons from minimal cognitive impairment patients. Minimal cognitive impairment patients are those that present, as the name says, some degree, but not a high degree of cognitive impairment, and sometimes progress to Alzheimer's disease. Now, both in MCI and in Alzheimer's disease, you can see quite a number of neurons expressing cyclin D or PCNA, which are both markers of active cell cycle. In controlled neurons from healthy individuals, you never see those. So that means that in these conditions, some neurons are capable of re-entering the cell cycle to some degree. And in fact, again, in the last few tens of years, we have learned that neurons have a peculiar correlation between reactivation of the cell cycle and cell death. Very strangely, neurons reactivate the cell cycle before dying. And in fact, in many cases, reactivation of the cell cycle is a requisite for dying. If you block the cell cycle, neurons don't die, even in the presence of the noxious stimuli that would kill them otherwise. So, but this is typical of neurons. Nonetheless, this evidence here, and many others, of course, show that neurons can re-enter the cell cycle, although partially, usually when they are going to die. Another example is shown here. This is skeletal muscle infested by Trichinella spiralis, which is a parasite of muscle that triggers nuclei in fibers. Here are nuclei from this fiber, 1, 2, 3, 4. These are nuclei from another fiber that runs like this. Another fiber is here. And when you see granules, black granules on these nuclei, it means they have incorporated 3-sheated thymidine, which is an old marker showing DNA synthesis. So it has been shown that muscles infested by Trichinella spiralis reactivate DNA synthesis and indeed arrest them at the end of DNA synthesis before dividing. Nonetheless, again, skeletal muscle is a classical example of terminal differentiation, yet the nuclei of these cells, of these fibers, can reactivate the cell cycle and even replicate DNA. One last example is podocytes. Podocytes, shown here by these arrowheads, are cells that contribute to make up glomeruli, kidney glomeruli. Glomeruli have long been considered unable to repair themselves. Once you have lost the glomerulus, it's lost forever. That comes basically from clinical observations. It is very rare or even impossible to recover from chronic renal failure. Nonetheless, this evidence shows here that podocytes, the precursor cells for glomeruli, do reactivate the cell cycle in pathological conditions. When glomeruli suffer damage, podocytes are reactivated. They express cyclin A, they express Ki67, again, markers of the cell cycle. So I'm saying all this to introduce the concept of regenerative medicine. What we're trying to do is to understand what makes a terminally differentiated cell decide to exit the cell cycle, and what makes it reaffirm, it makes it confirm this decision so that it never re-enters the cell cycle ordinarily. So what has this to do with regenerative medicine? Well, in the science fiction literature of the 50s and of the 60s, you often encounter mysterious rays, such as these, that can heal any kind of wounds, any kind of diseases from the outside of the body. Well, what I'm proposing here is that, in fact, we can, in principle, heal many diseases, many traumas of the body, or we could, I should say, we could, if we were able to reactivate terminally differentiated cells in a safe, controlled, reversible fashion. Imagine if we could reactivate proliferation of neurons. Imagine if we could speed up, accelerate, promote proliferation of cardiomyocytes or of the cells that make up the endocrine gland, many endocrine glands. These are all tissues and systems that cannot replace lost cells because their parenchyma is composed of terminally differentiated cells, and because they do not have a stem cell compartment sufficient to guarantee, to allow efficient replacement of lost cells. So if we were able to induce terminally differentiated cells to proliferate in the way I've just said, we would be able to repair, potentially, we would be able to repair lesions that are now unrepairable. And if this, again, looks like science fiction, I'd like to introduce this animal to you. Some of you might know it. It's the newt, in Italian tritone. A newt is a small animal, few centimeters in length. It's an amphibian, and it's well known for its amazing ability to regenerate any part of its body. By any part, I really mean it. You can not only cut off its tail or its limbs or any other external part of the body. You can cut off any part of internal organs, including, say, the tip of the heart, the spinal cord, the brain, an eye. They will all regenerate and go back to the original state. Provided, of course, that you don't kill the animal by the amputation itself. The animal must survive in order to regenerate. That's, and of course. Now, to show you the amazing ability of these newts to regenerate, here is a time series of photographs in which the anterior and the posterior limb of a newt have been amputated. And you see the stump here of the anterior and the posterior limb, and then you see what becomes of them in a matter of a few weeks. You see the limbs regrow, And although this series here is interrupted, they will go back to exactly the same size and shape of the original limbs. Now, I'd like to point out that newts are not strange, unicellular, or very simple organisms that are little more than a bunch of cells. Newts are vertebrates. They're much like like ourselves. You know, they have the same organs, systems, tissues, and in fact, you can draw many parallels between them and us. But there are things that distinguish newts from mammals and from humans in particular. And so people have investigated, we still are investigating how they perform these amazing feats of differentiation, of regeneration. And here is an interesting piece of evidence that shows how an amputated limb is regenerated. What you see here is a single myofiber, a muscular fiber, in a limb, which has been injected with a red fluorescent stain so that we can follow this individual fiber. The limb has been then amputated in the proximity of the fiber. And here is what happens in a matter of a few days. Initially, the fiber doesn't seem to react to the amputation, but then it splits into a number of fragments, which are in fact mononucleated cells. You know that muscle fibers are multinucleated, they are syncytial, but it splits into mononuclear cells, which then divide, and then eventually redifferentiate and contribute to the regeneration of muscle and other tissues and other cell types. So this is something that newts can do and we cannot. And so what we in my laboratory do is to try to understand what's the difference between animals such as the newts and us, and how terminally differentiated cells exit the cell cycle. What are the molecular mechanisms underlying exit from the cell cycle, and what are potential mechanisms capable of bringing the cells, the terminally differentiated cells, back into the cell cycle, eventually for regenerative medicine purposes, as I've said. So what we mostly work on in our laboratory is skeletal muscle. Skeletal muscle, we use it because it's an excellent example of terminal differentiation. Aside from infestation from Trichinella spiralis, there are no other known examples in nature of skeletal muscle cells re-entering the cell cycle. And these cells are absolutely resistant, refractory to a large number of attempts to reactivate their cell cycle. They are also relatively easy to work with in the laboratory. They can be grown, propagated as myoblasts. There are many myoblast cell lines, immortalized cell lines that you can grow limitlessly. Or you can actually put in culture primary cells from muscle biopsies from animals or even from humans. We have a lot of muscle, taking a small bit of it doesn't harm anybody. So once you have grown myoblasts in the desired quantity, you can easily induce their differentiation by withdrawing growth factors, usually represented by serum. Fetal bovine serum is the most common source of growth factors in the laboratory. So if you remove growth factors, myocytes will stop proliferating. Sorry, myoblasts will stop proliferating, will become myocytes, that is already terminally differentiated cells, which are still mononuclear. They have a single nucleus in each cell, which then will eventually fuse into syncytial multinucleated structures called myotubes. Now, in the skeletal muscle, but not necessarily in other terminally differentiated cell types, exit from the cell cycle is a prerequisite for differentiating. Myocytes must first arrest the cell cycle, then they can begin expressing muscle-specific cells represented here by the red stain. So myocytes are as terminally differentiated cells as myotubes. This is what we work with. And this is what actual myotubes look like in culture. This is the schematic I've just shown you, and these are actual myotubes. You see elongated structures in which you can almost see nuclei, at least in some parts. These are nuclei. You see that some of these structures are branched. Some are triangular. Here you see more branches. And the smaller cells are contaminating myoblasts. It is virtually impossible to get a culture of pure myotubes. You always have some degree of contamination by myoblasts, which is a small complication in dealing with these cells, but we are able to work with them anyway. So, is it clear so far? Are there any questions?
Everything is clear.
Good, thanks. Everybody agrees?
Yes, thank you.
Okay, so these, as I've said, myotubes are very resistant to a number of stimuli that would push non-terminally differentiated cells to proliferate. Say they are resistant to all growth factors people have tried. They're resistant to a number of oncogenes that are strong inducers of proliferation in non-terminally differentiated cells. So at some point, a long time ago, in fact, we wondered whether these cells were totally unable to respond to proliferating stimuli, or they were able to, in some way, to sense them and to give a limited response to them. In order to investigate this issue, we did a simple experiment, which was nonetheless very fruitful because it gave us precious information. What we did is shown here. We compared myotubes, postmitotic myotubes, with quiescent myoblasts. Quiescent myoblasts are just temporarily arrested and can go back to the cell cycle when properly stimulated. So we stimulated both myotubes and myoblasts with 10% serum, the source of growth factors I've already talked about. And then by Northern blot, these are Northern blots, we investigated the expression of a number of genes at various times after the beginning of the stimulation. So here we are at the very beginning of the stimulation with serum. Here we are half an hour from the beginning, 1 hour, 2 hours, 4 hours, and so on. And what we found was very neat, very clear. In the first several hours, the responses of myotubes and myoblasts are indistinguishable. We have a very early and very short, sharp peak of expression of C-FOS, here and here. We have 2 peaks of expression of C-Jun. We have 2 less well-defined peaks of expression of CMYC, 1, 2, 1, 2. And we have, once it is initiated, the expression of ID1 or cyclin D1 stays on in both cell types. What differentiates the two cell types is the late response, say beginning from 8 hours of serum stimulation. The genes expressed in late G1, such as cyclin E, cyclin A, PCNA, are strongly upregulated in quiescent myoblasts, while they're not upregulated in myotubes. The faint signal you see here is due to the contaminating myoblasts I was alluding to before. Now, these genes, cyclin E, cyclin A, PCNA, and many others, mark essentially the transition between G1 and S phase, because these cells indeed are undergoing DNA replication. They're entering S phase, while these myotubes are not able to enter S phase because they never upregulate those regulators and effectors that are necessary for DNA replication to occur. So let's put these data on a linear schematic and partial schematic of the cell cycle, which regards the cell cycle as a linear series of events. Cells are standing in G0. When stimulated, they enter G1 and continue on to S phase ordinarily. But if they're myotubes, they're stimulated by growth factors and respond much like myoblasts in the first part of G1, but then hit a wall, hit an obstacle that they are unable to pass, which prevents them from going through the second part of G1. Of course, we were very interested in the nature of this obstacle, but at the time had no idea what it was. However, at the same time, we were looking for ways to reactivate the cell cycle in terminally differentiated cells, as I've said. And we had reasons from the literature and from our reasoning to believe that an oncogene called E1A, which is an adenovirus oncogene, would be able to reactivate terminally differentiated myotubes. Now, it is a bit long to explain why we thought so, but we did an experiment. It was an easy experiment because adenoviruses can infect a large number of cells. They have a broad host spectrum, as virologists say. So it is easy to infect cells in culture with adenoviruses. Adenoviruses then spontaneously express E1A into the infected cells. And this is the result. These are immunofluorescence in black and white pictures. These are old experiments. We stain myotubes with an antibody against myosin heavy chain, a marker of terminal differentiation in skeletal muscle cells. We stain the nuclei with DAPI, which is a general marker of DNA, which beautifully shows the nuclei here. And then also we stain the nuclei of the same myotubes with an anti-BUDR antibody. Now, BUDR, bromodeoxyuridine, is a molecule similar to thymidine, which gets incorporated into nascent, into newly synthesized DNA instead of thymidine, if of course you add BUDR to the culture medium. So being positive for BUDR shows that you have recently synthesized DNA. And as you can see, the myotubes infected with adenovirus and expressing E1A indeed re-enter the cell cycle, synthesize DNA. All of the nuclei of this myotube here are BUDR positive, and they are all of the nuclei visible in this picture. So all of them have re-entered the cell cycle. Same here for this other myotube. All of the nuclei are BUDR positive. And here you have an occasional binucleated small myotube, which has not incorporated BUDR, showing that we don't have a 100% reactivation efficiency, but we're close to it, in fact. So if we, when we did an experiment similar to the serial northern blots I've shown you, we got an interesting sort of mirror result compared to serum stimulation. Whereas serum stimulation was unable to take the cells beyond a certain point in mid-G1, E1A directly stimulated genes close to the G1S transition, actually jumping from G0 to this point. Now, this was very satisfactory because we had found an efficient, simple, and very effective way to reactivate the cell cycle in terminally differentiated cells. It was basically a first. However, we didn't necessarily like E1A in all regards. Well, first of all, because E1A could not give us any information about the block, because it didn't force it, it didn't break it. It just jumped over it. And so we still didn't know what the block was made of in these cells. Also, E1A is an oncogene, and in the even in the distant prospect of using this oncogene of reactivating terminally differentiated cells in human therapy, of course, using an oncogene is not desirable. So we looked for cellular as opposed to viral genes capable of reactivating the cell cycle in terminally differentiated cells. And by scoring the literature by trial and error, we found that the combined expression of cyclin D1 and CDK4 could reactivate the cell cycle in myotubes. And because the adenoviruses had worked so well in the previous experiments, we devised two recombinant adenoviruses, one expressing cyclin D1 and another expressing CDK4. Now, as you probably know, the cell cycle is regulated by various combinations of cyclins and CDKs. This particular one, cyclin D1 and CDK4, is mainly active in the early G1. Here is the experiment. We infected myotubes here, these two lanes, with either the two recombinant adenoviruses expressing cyclin D1 and CDK4, or control viruses. And here are spontaneously proliferating myoblasts. You see that we got huge overexpression of both cyclin D1 and CDK4, and the outcome of this overexpression was reactivation of the cell cycle in the infected myotubes. Here again, you see in red myosin heavy chain, marker of differentiation, and in green, you see BUDR incorporation, a clear sign that these myotubes had replicated their DNA. Control myotubes, of course, did not incorporate BUDR. And this graph here shows that the myotubes entered efficiently. More than 70% of the myotubes, in fact, re-entered as phase, and rather synchronously, within a few hours, re-entered the cell phase, so that, again, this graph shows that our method is efficient and that the myotubes re-enter the cell cycle synchronously so that it is easier to analyze them by biochemical and molecular biology means. Because if the cells behave in the same way at every single moment, you can analyze the whole culture and regard the culture as mirroring the behavior of each individual cell. And also we showed that cyclin D1 and CDK4 overexpression could reactivate not only myotubes, but also other terminally differentiated cells such as adipocytes. Here we see mitotic adipocytes. These are their nuclei. And even neurons. Here are neurons incorporating BUDR following expression of cyclin D1 and CDK4. Now, this was all good, but we needed this, as I've said, this huge overexpression of the two proteins to reactivate the myotubes. So one problem we had in mind was that perhaps this was a totally artificial situation that we made up in the laboratory, which had nothing to do with the physiology of these cells. So a laboratory trick that told us nothing about the physiology of the cells. However, we got a surprise when we measured the kinase activity associated with these high levels of the two proteins. Consider that as far as the cell cycle is concerned, it's not really the amount of cyclin D or CDK proteins that matter. What matters is the kinase activity, the enzymatic activity that is expressed by these proteins. So when we measure the kinase activity of these proteins in the myotubes, we found, to our surprise, that the amount of kinase activity in the overexpressing myotubes was equal to that found in spontaneously proliferating myoblasts. So, by going back to our schematic, it showed that this wall here, this obstacle here that prevented progression of the cell cycle beyond MAGE1 was just the lack of CDK4 activity. When we reconstituted physiological amounts of CDK4 activity, then the cell cycle was allowed to progress. And that told us what this obstacle was all about. Now, we still had a paradox. Why did we need such a larger expression of the two proteins in order to obtain a physiological amount of CDK4 kinase activity? Well, we devised a model to try and address this question. The model went like this. In a non-terminally differentiated cell, of which this is the plasma membrane and this is the nucleus, when a growth factor binds to its receptor, it activates a signaling cascade. This is a very simple schematic, of course. Things are more complicated than this. But let's get simple. It activates a signaling cascade that goes to the nucleus and triggers expression of cyclin D, of a cyclin D, one of the three family members of the D cyclins. Cyclin D combines with CDK4, which is more or less constitutively expressed in all cell types, and that allows the cell cycle to proceed. Now, the model went in terminally differentiated cells. an inhibitor or several inhibitors prevent cyclin D and CDK4 from activating. And so myotubes cannot respond to growth factors, at least not completely, as I've shown you. So a model is something we very often use, not only in science, but even in our regular life, in everyday life, I should say. We use models because our minds are not capable of considering thousands of factors, of elements, when thinking about whatever natural phenomenon. They can only consider a few elements at a time. So we use the simplest possible model to think about our problems. And in this case, you see, there are very few elements in this model. But as any useful model, this model made crucial predictions. The model said that if we could identify these or these inhibitors, and we could then remove them from the myotubes, we would reconstitute the ability of myotubes to respond to growth factors, which was at the time the holy grail in the field. What could these inhibitors be? Well, the literature was very confusing at the time. So we resorted to an unbiased experiment, an experiment that did not require previous assumptions or knowledge. We said, let's look for anything that can prevent CDK4 activation. How did we do that? We moderately overexpressed cyclin D1 and CDK4 in the myotubes. Moderately means just below the threshold necessary to reactivate the myotubes, just below the amount that would trigger reactivation, cell cycle reactivation in the myotubes. Why we did that? Because we thought that in this way, cyclin D and or CDK4 would bind to all the available inhibitor in the myotube. And so we had the highest chance of precipitating the inhibitor together with CDK4. And we set the conditions to do the experiment without reactivating the myotubes, which would have reactivated the whole cascade of the cell cycle and complicated very much the interpretation of the experiment. So here's what we did. We, as I've said, we overexpressed cyclin D1 and CDK4. We lysed the cells, we broke them up, and we immunoprecipitated the protein extracts from these cells with an anti-CDK4 antibody. We did an immunoprecipitation with this antibody, and then we looked for proteins co-precipitated with CDK4, proteins that in vivo in the cell bound CDK4 and thus were precipitated together with CDK4 by an anti-CDK4 antibody. And then eventually we identified the co-precipitated proteins by means of mass spectrometry. So here is the actual experiment. Here is the anti-CDK4 lane. This is a gel separation of proteins stained with silver stain. So we are seeing all proteins present in this lane. And this is a control immunoprecipitation done exactly in the same way as this one, except we used an irrelevant antibody. An antibody that bound a viral protein not present in these cells. So in theory, it should have precipitated nothing. But in reality, we know that immunoprecipitations are always dirty in that many proteins precipitate non-specifically. They bind to the antibody in a non-specific way. They bind to the beads that we use to carry out immunoprecipitations, again in a non-specific way. And so we have to have ways to distinguish non-specific precipitation from specific co-precipitation. The way we do it is by comparing these two lanes. Whatever is present in both lanes is most likely background, non-specifically precipitated proteins. Whatever is present in this lane here, but not in this other, is presumably a protein co-precipitated along with CDK4. And so you can see a few of these bands here, 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 etc. And so we identified the proteins present in several of these bands. Here is part of what we found. This band here contained cyclin D1, 2, and 3, the whole family, and CDK4. These proteins have similar molecular weights, though, so they migrate at the same point in the gel. This protein here contained PCNA. And the most interesting protein was this one, because this band contained P21. P21 is a known cell cycle inhibitor and the only one found in this experiment. So probably you are guessing what we did next. We suppressed the expression of P21 by siRNA, by RNA interference in myotubes. Here is P21 levels expressed in control myotubes, and this is after interference. You see, there was deeply downregulated by the interference. And to our surprise, when we did this experiment, well, before telling you what we found, I'll tell you what the prediction, I'll remind you of what the prediction was. The prediction was by removing the inhibitor or the inhibitors, we would reconstitute the ability of myotubes to respond to growth factors. But we thought that because the myotubes are so stubborn, they are so refractory to all attempts to reactivate their cell cycle, that they had a redundant network of different kind of breaks that kept them from re-entering the cell cycle. We thought that P21 was perhaps just one of them, and that by removing P21, we would have simply facilitated the cell cycle re-entry in myotubes. Basically, we thought, well, if we remove P21, we are probably going to need lower levels of cyclin D1 and CDK4 to reactivate the cell cycle. But instead, what we found was that as soon as we removed P21, the myotubes spontaneously re-entered the cell cycle. Now, why was that so surprising? Because we didn't add any growth factors in this experiment. So these myotubes here were not responding to growth factors. They re-enter the cell cycle in the absence of any external stimuli. This went beyond our predictions, and so required us to change our model, to switch to a different model. Now, models in science and elsewhere are only useful as long as they adhere to reality. If they behave like the natural phenomenon they model. If they don't, you should throw away as soon as possible and make a better model. That's what we did. But of course, models, you have to elaborate the model. You don't necessarily have the information required to make a new model. So how did we do that in this particular case? Well, we went back to our immunoprecipitation. Sorry. I always forget at this point. I don't know why. This picture shows that that cyclin D1 and CDK4 not only reactivated DNA synthesis, but also brought the myotubes all the way to mitosis. These are horrible mitosis, but I think you can see the condensed chromosomes and the more or less individual nuclei here, 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 and here. These are in fact mitotic catastrophes. I'll tell you later why they're catastrophes. But anyway, this picture here shows that the cell cycle reactivation in myotubes is complete. They not only replicate their DNA, they go all the way through mitosis, and in fact, they even try to divide. But then again, we were left with the problem of understanding the unpredicted behavior of these myotubes. And so we went back to our immunoprecipitation and noticed that, among other things, together with CDK4, cyclin D3 was immunoprecipitated, was co-precipitated. Now, we had overexpressed cyclin D1, not D3. So cyclin D3 must have come from the cells. And so this precipitation here suggested that cyclin D3 CDK4 complexes might be responsible for the reactivation of the myotubes. Why cyclin D3 and not cyclin D2? Because we remembered data in the literature that said that in myotube, that showed that in myotubes, there are multi-protein complexes that, among other proteins, contain cyclin D3, CDK4, and P21. Once again, I'm not showing entire complexes. There are other proteins in the myotubes that make up these complexes. But these are the important parts. Again, we devised a model that took into consideration only the elements that we thought were important for the phenomenon we were studying. And so we disregarded the other proteins and concentrated on cyclin D3, CDK4, and P21. Again, as a good model, this made a prediction. And this was a very strong, clear-cut prediction. It said, if and only if it is true that cyclin D3 and CDK4 are responsible for the reactivation of the myotubes upon removal of P21, then on removing P21, we should see reactivation of the myotubes. But if we remove P21 and cyclin D3 at the same time, or P21 and CDK4 at the same time, we shouldn't be seeing any reactivation. So we did the experiments, and I'm not going through these plots, which I'm showing just because, biochemically, it all worked as expected. What's important in this slide is the results. And here are the results, very clear cut. Of course, if we interfered with control siRNA, no myotubes re-entered the cell cycle. But if we interfered P21 here alone, 68% in this particular experiment of the myotubes re-entered the cell cycle, synthesized DNA. But if we interfere with P21 and cyclin D3, then this number went down to basically zero. And if we interfere with P21 and CDK4, again, the number dropped to zero. So that validated the model and said both cyclin D3 and CDK4 are absolutely necessary for the reactivation of the myotubes that follows P21 suppression. Now, that again was a milestone to us because it told us what prevented the myotubes from ordinarily activating CDK4 kinase. Remember, I told you that the block here in MYT-G1 was merely due to the absence, to the inability of myotubes to express CDK4 kinase. Now we had learned why this kinase was unable to be activated, and that was because of the high expression of CDK inhibitors, most importantly P21, which prevented the kinase from being activated. The experiment I've shown you also explained very satisfactorily why we needed to express such high levels of CDK4 and cyclin D1 as I've shown you. That's because we needed to titer out the inhibitors. We needed to remove them, to sequester them, until further expression of CDK4 and cyclin D1 were now able to form active complexes. In addition, it explained why the myotubes were able to re-enter the cell cycle in the absence of growth factors. That was because growth factors are needed for two reasons. First, to induce expression of cyclin Ds. Second, to greatly facilitate the assembly of cyclin D CDK4 complexes. But those complexes are already present in the myotubes. So all we were doing by suppressing P21 was allowing already pre-existing cyclin D3 CDK4 complexes to activate and drive the cell cycle. Now, this is a bit complex, so if I haven't been able to be clear, please let me know.
I have to say I lost one of the growth factors reasons why we need them. One is to... Thank you.
Okay, let's go back to the schematic here. Let's say growth factors are needed for two reasons. One is to drive cyclin D expression. Cyclin Ds, especially cyclin D1, are driven by growth factors. They're expressed only in the presence, or mostly in the presence of growth factors. So our initial model did not predict that cells would re-enter the cell cycle in the absence of growth factors because it did not predict that it would have any cyclin Ds. Also, it is known that cyclin D CDK4 complexes only assemble efficiently in the presence of growth factors. And so they should have been required for that too. But neither of those activities, in fact, require growth factors in the myotubes because cyclin D3 expression doesn't need to be driven because cyclin D3 is already there at high levels in myotubes. And because the complexes are already assembled, and so they don't need facilitation for their assembly. Is it better now?
Yes, it is. Thank you.
You're welcome. So good. So far, we were very happy of our results, except for one major problem that I haven't told you about yet. And that is, whatever we did, however we reactivated the myotubes, we could not obtain true proliferation. We could not make these myotubes to really proliferate and make more cells. And that was one of our main long-term goals. Remember, one of our goals is to use what we learn in regenerative medicine. What I've told you so far is that we are able to reactivate the myotubes in several ways. We can express C1a, we can overexpress cyclin D1 and CDK4, or 6, they belong to the same family. We can suppress cyclin-dependent kinase inhibitors, basically P21 and some others, depending on the cell type. All of these practices reactivate the cell cycle in the myotubes, but none of them elicits real proliferation. Because E1A induces apoptosis in the reactivated myotubes. Myotubes reactivated by cyclin D1 and CDK4 arresting the first G2 after DNA synthesis, so they never reach mitosis, basically. And when we suppress P21, we have either mitotic catastrophe, as I've shown you in one picture, or apoptosis. Now, this looks like a bit of a mess, meaning we do different things and we obtain different outcomes. So at first sight, you might think, well, you haven't yet found the right way to reactivate the myotubes. You do different things, you obtain different effects. None of them is what you desire. So try another way to reactivate the myotubes. But we thought, well, perhaps all these failures, if you want to call them so, all these failures perhaps have something in common. Remember, differences in natural phenomena are infinite. What scientists look for is regularities, commonalities. So we were looking for a common mechanism explaining all of these different outcomes. It took us quite some time, but eventually we took advantage of an important observation I've not told you about yet. That is, of course, post-mitotic cells are not the only kind of non-proliferating cells. Quiescent cells, reversibly growth-arrested cells, are also in G0, much like post-mitotic myotubes. Senescent cells are also arrested in G0, again, classically irreversibly arrested in G0. But we showed that by removing CDKI, cyclin-dependent kinase inhibitors, from these cells, we could reactivate all of them. And that was a major finding in itself. However, there was an important difference between myotubes and the other cell states or cell types. When we reactivated quiescent cells, they really proliferated. These are fibroblasts reactivated by P21 suppression in the absence of growth factors. And they doubled in number in 3 days. These are control cells that basically did not proliferate at all. And even if we took senescent cells, they tripled in 3 days by removal of P21. They increased somewhat by removing another inhibitor, P16. And there was no proliferation whatsoever, of course, in the controls. So these cells effectively proliferated. And by studying them, we learned and we know that they do not suffer any damage. But when we reactivate the myotubes in the very same way, as I've shown you, they basically die off. So this means that there is nothing wrong with the means we use to reactivate the cells. The problem is in the myotubes. It's intrinsic to the myotubes. So what's wrong with the myotubes? Well, after some time, we've said, we finally found an answer. And we found that myotubes do undergo DNA replication, but they never complete it. You can show it, once you have understood that, you can show it in many, many ways. This is the most pictorial one. Oh, this shows essentially, let's talk about this for a moment. This picture shows that when we reactivate quiescent fibroblasts, for example, mitosis are regular. This is a nice anaphase. There's nothing to say about it. If you reactivate myotubes in the same way, again, they undergo mitotic catastrophe. What is a mitotic catastrophe? It's basically an extreme fragmentation of the chromosomes, which also brings about a large number of problems in the correct segregation of chromosomes. If they are fragmented, not all of the fragments, most of the fragments won't have a centromere, so they will be unable to segregate, pulled by the mitotic spindle. So eventually these, of course, kill the cell or the daughter cells if they form at all. So again, the difference was in the cell type. The myotubes would not proliferate in the presence of the same stimuli that would make non-terminally differentiate cells to proliferate. And the reason was, as I've said, that they cannot complete DNA replication. This is one way to show it. This is a DAPI staining of the DNA in a single metaphase nucleus in a myotube. It is ugly because this is a mitotic catastrophe, but I think you can see the metaphase plate here and the chromosomes forming stokes irradiating from the center of this metaphase plate. This myotube has been incubated throughout the cell cycle with BUDR, so it has incorporated BUDR into its DNA. And because it's always been in the presence of BUDR throughout the cell cycle, you would expect all of its DNA to be uniformly, as we say, substituted by BUDR, meaning that BUDR has replaced thymidine. But if you superimpose the two images, you see clearly that not all of the DNA contains BUDR, which means that this myotube has entered mitosis without having replicated completely its DNA. So this leads us to our current model. When you reactivate whatever non-terminally differentiated cell, such as a fibroblast, the DNA is replicated completely, and at mitosis, sister chromosomes are separated as normal. If you reactivate myotubes, DNA replication is incomplete, which, depending on the means you have reactivated the cell cycle by, and I won't go into the biochemical details because it would take long, but we can explain why, depending on these means, either the myotubes die by apoptosis, or if they can make it out of S phase, they arrest in G2 because the G2 checkpoint is activated by the presence of damage in the DNA and by the presence of incomplete DNA replication. And if they force this block and enter M phase, then sister chromatids cannot separate because DNA replication is incomplete. And of course, there is DNA fragmentation, and so this altogether defines a mitotic catastrophe. And so the question became, what is the molecular nature of the obstacle to full DNA replication? Now, this is a damn question. This is one of those vague questions that science has such a hard time addressing. This is too vague. Whatever in a myotube, which is as complex as any cell type, it has thousands of proteins, thousands of macromolecules, a huge number of organelles. It's so complex, and you are asking, what prevents DNA replication? Who knows is the instinct answer. So we thought we needed a way to reduce this question to something more tractable. And so we divided the potential answers into two classes. We thought DNA replication can be hindered by either functional obstacles or by structural problems. Now, by functional obstacles, we mean whatever doesn't function properly in the myotubes at the molecular biochemical level. Think about a gene that is not expressed, a protein that is not translated properly, an enzyme that is segregated into an improper compartment, intracellular compartment, a post-translational modification that is not executed at the right time, on the right amino acid, whatever you can think that doesn't work well in the myotubes and that can hinder, can arrest, impede full DNA replication. There is another class of potential problems, and that's structural. We thought of this possibility that the chromatin of myotubes might be itself physically responsible for the inability of DNA polymerase to complete DNA replication. It could be at least partially impervious to DNA replication. It could be physically, structurally hindering DNA replication so that it could never be completed within a single S phase. So how did we address these two possibilities? Well, let's start with the most obvious one, functional obstacles to DNA replication. For a long time, we searched for such obstacles, but as much as we could compare myotubes with myoblasts or other proliferating cell types, we did not find any problems as far as we could look for them in the myotubes. Until eventually we thought of something. We thought of the nucleotide pool. Now, what is the nucleotide pool? It's those nucleotides that are free-floating in the cytoplasm and that are ready for DNA synthesis to take place. Basically, they are the building blocks of DNA. Cells constantly have a pool, but this pool is very expensive, energetically speaking. So the cells usually have a small nucleotide pool, and they only expand it at the time when they replicate DNA. Outside of the S phase, the nucleotide pool is small, and it's only needed for replicating mitochondrial DNA. You know, mitochondrial DNA is replicated independently of nuclear DNA. And to some extent to repair DNA. When you need to replace a few nucleotides or short stretches of DNA, you need nucleotides. You need a nucleotide pool, but that's a very small part what it is necessary for. So non-proliferating or non-S phase cells have a small nucleotide pool. We thought that myotubes perhaps were unable to expand the pool, or they were not able to do it in time to follow DNA replication. That might prevent DNA replication from completing. So we measured together with our collaborators at the University of Padua. This is a very specialized kind of measurement that we were not able to perform. We measured the nucleotide pool in myotubes, which, as expected, was, here are the myotubes, much lower, much smaller than what found in proliferating myoblasts. You see, each one of the nucleotides has its own level. They are not present in equal levels, first of all, because they are not represented in equal levels in the DNA. Second, because they have other functions other than being the building blocks of DNA. And so their reciprocal levels are tightly regulated, and they always are in the same ratio with one another. So this is what a regular, a proliferating nucleotide pool looks like. This is what it looks like in myotubes. So now we asked, what does the nucleotide pool in myotube, in a reactivated myotube, look like? It looks like this. You see, two of the nucleotides, CTP and ATP, are highly overexpressed, say 28, 30, or 36 hours after reactivation by suppression of P21 and P27. P27 is another cyclin-dependent kinase inhibitor. We thought about this large overexpression of these two, large overrepresentation of these two nucleotides, but we thought, for theoretical reasons, that it wasn't so important. What was important was that the levels of TTP and GTP were much lower than found in proliferating myoblasts. And perhaps they were insufficient to allow full DNA replication. So following this hypothesis, DNA replication did not complete merely because there were not enough building blocks. Just to make sure things were working properly with our measurements, we measure the DNA pool, the nucleotide pool in quiescent fibroblasts. And you see it is even smaller than in myotubes. And we reactivated these fibroblasts and found that the nucleotide pool was quite similar to that of proliferating myoblasts. So everything was all right. So by hypothesizing that two nucleotides were present in too small amounts to allow full DNA replication, we decided to try and replace them. And so we provided nucleosides, the precursors of nucleotides, from the outside. Nucleosides are capable of crossing the plasma membrane, so you can give them from the outside. Nucleotides cannot cross the plasma membrane. That's why we use nucleosides. But they are rapidly converted into nucleotides in the cell by simple kinase activities. So that's a perfect way to replace the nucleotides. And what we found is that in proper conditions, we could reconstitute levels of TTP and GTP that were quite close to natural levels in myoblasts. Now, what was DNA synthesis in myotubes reactivated in the presence of nucleoside supplements? Okay, first of all, immunofluorescence evidence. These are myotubes reactivated without supplementation. You see that BUDR incorporation is rather pale, and most important, is very irregular. You see patches of brighter BUDR incorporation in the nuclei, and patches that are quite dark. And within the same myotube, some of the nuclei are quite dark. In fact, basically negative for BUDR. They did not appreciably synthesize DNA. Now look at this other myotube in which we supplemented the nucleotide pool. First of all, the nuclei are brighter. Most important, they are even. Fluorescence is much more uniform within the nuclei and among different nuclei. So it suggested that DNA replication was enhanced. And indeed, when we measured DNA replication, we found that it was. So here we measure the amount of DNA by microfluorimetry, a technique I'm not going to explain unless some of you is interested. We measure DNA content in each nucleus. And by counting enough nuclei in different myotubes, we found these results. Okay, these are quiescent myotubes, non-reactivated myotubes. All of their nuclei have the same DNA content, which on an arbitrary scale is what? About 19, okay? This is what they do when you reactivate them by simply removing P21 and P27. So the DNA curve shifts to the right. The amount of DNA, the average amount of DNA per nucleus is indeed increased. DNA has been synthesized, but it's little more than naturally present in non-proliferating nuclei in myotubes. If we supplement the nucleotide pool, then the shift to the right is much more marked, much more evident. And the average content of DNA per nucleus is much larger, is about 30. Nonetheless, so this shows that supplementing, giving myotubes extra nucleotides really makes a difference, really allows them to synthesize more DNA. And so they're in fact limiting, their amount is limiting for DNA synthesis. However, it also shows that it's not sufficient to expand the nucleotide pool to allow full DNA replication, because even with supplementation, the average content of the nuclei is 30, which is not twice as much as 19. So even in these conditions, myotube nuclei did not complete DNA replication. And in fact, they did not survive cell cycle reactivation, even in the presence of supplementation. Nonetheless, we had found a functional reason, though partial, for lack of completion of DNA synthesis. What else could be there? Well, the other possibility, as I've mentioned, was that there were structural problems in the physical problems in the structure of chromatin, which was suggested, among other things, by the patchy nature, this uneven, non-uniform incorporation of BUDR, as if some parts of the chromatin were efficiently replicated and some others were not. So we set out to determine what parts of the chromatin were more easily replicated than others. We thought, well, these bright areas here are probably easily, efficiently replicated, And the darker areas here are not. So let's see what are efficiently replicated, what areas are efficiently replicated and what are not. Because the ones that are not replicated or are less efficiently replicated contain the problem. And we will be able to look for the problem in those areas. To do that, we reactivated myotubes, labeled them with BUDR. Again, BUDR incorporation was uneven, patchy. Then extracted the DNA from the myotubes and separated the newly synthesized DNA by an ancient technique called cesium chloride gradient. Again, I don't have time to explain how this technique works, but if you're interested, I'll be happy to tell you. So let's just assume, as we did, that we isolated newly synthesized DNA. So that mirrored the areas that were efficiently replicated, but could not contain any DNA that was not replicated. Is that clear?
Yes.
Okay, thank you. I'm sorry, but I cannot see you, so I cannot tell from your faces whether or not you are understanding me. So please do answer when I ask. So we took this DNA here and hybridized it to genomic microarrays, so that whatever was present would give us a signal in some areas, and whatever was not present, was not replicated, would not give us a signal. And we would be able to tell where was the problem. And here is the results we got. Basically, what you're seeing, the small dots are individual points in the microarrays. And the continuous line here is the average of the signal signals present in those dots. And as you can see, this line here oscillates around the 0 line, right? That means there is no preference. All areas in a myotube are replicated with the same probability. Not all of them are replicated within the same nucleus, because DNA synthesis is always incomplete in each individual nucleus. But there is no preferentiality. No area is more or less difficult to replicate. It's just random. This was very disappointing at first, but then we thought of what this experiment meant. It meant that these myotubes reactivated in this way by P21 suppression replicate their DNA in the absence of DNA replication timing. Now, you probably know that DNA replication does not occur all at the same time. Some regions of DNA are replicated earlier during S phase. Some others are replicated later during S phase. What is replicated earlier and later is typical of a given cell type. Every cell type has its own pattern of early and late replication. Why is that? Because it correlates with gene expression. Chromatin is open where genes are expressed and is generally replicated earlier. Chromatin is closed when where genes are not expressed, and is generally replicated later. So because chromatin state is correlates with gene expression, that explains why the DNA replication timing is typical of a given cell type, because each cell of that cell type expresses the same genes and so has the same replication timing. Now, by comparison, this is the same experiment carried out with myoblasts. Clearly, you see that there are peaks above 0 and valleys and throws below 0. So this shows that myoblasts have a DNA replication timing, as expected, and that myotubes reactivated in this way basically do not. Now, this was interesting because nobody understands in depth replication timing. It's been studied and described phenotypically, phenomenologically. We know a lot about about how DNA replication behaves, what's early, what's late, what determines at the general, at the holistic level in the nucleus, what determines what is replicated earlier and later. But we do not know, essentially anything about the molecular determinants of DNA replication timing. Now, one problem is that DNA replication timing is universal. All cell types, except very early embryo cells, replicate DNA with a specific DNA replication timing. So basically, you don't have the classical experimental setting where you have or you do not have DNA replication timing, and you can ask, what's the difference? What's missing when I don't have a DNA replication timing? This is for the first time a cell system that can replicate with or without a replication timing. Why am I saying that? This is the same experiment carried out this time with next generation sequencing. It's conceptually the same, it's just technically different. And then again, you see that, for example, on chromosome 1, this is chromosome 1 of myoblasts. Some regions are replicated early, the red ones, and some regions are replicated late, the blue ones. And so there is a very specific pattern that is faithfully replicated when we reactivate myotubes by expressing E1A. You see what is blue here is blue here, and what's red here is red here, all over. When we suppress P21, basically there is no timing. Again, the timing oscillates around 0. There is no preference in whatever is replicated earlier or later on whether something is replicated or not replicated at all. Same probability all over the chromosome and all over the genome. So again, now we have a system which has or has not DNA replication timing, and we can ask interesting questions at the molecular level. And in fact, we are doing that. But then again, we are back to our main question. So is there a structural obstacle to full DNA replication? And if it is there, what is it? So we thought of an experiment that would show if there is a structural problem to DNA replication in myotubes. We thought of this experiment. Let's say we isolate nuclei from myotubes. These are non-reactivated myotubes, just regular myotubes. Let's say we isolate nuclei from them and we put them into a replicating extract of Xenopus eggs. Now, what is it? Well, Xenopus, as you know, is a frog. If you take a female frog and you squeeze it as gently as possible, the frog doesn't like it, but it doesn't hurt it too much. You get a lot of eggs. You squeeze them out of the frog. And if you break the cells open, spin down, centrifuge down whatever is insoluble, and you eliminate these debris, whatever is left in solution is an extract of the eggs, which contains all the necessary ingredients to replicate DNA. It is so powerful that you can put naked, pure DNA into it, and it will replicate it. So it contains just everything that's needed to replicate DNA. So it's completely independent of whatever nuclei or DNA you put into it. Okay, so there are two possible outcomes of this experiment. When you put nuclei from myotubes into this extract, either they undergo complete DNA synthesis, or DNA synthesis is still incomplete, though in the presence of all the necessary ingredients to ordinarily carry it out. So, in other words, if the outcome is this, complete DNA synthesis, then probably the only difficulty in myotubes is biochemical, is functional. When you have the necessary ingredients, some of which probably are missing in the myotubes, DNA replication is complete. On the other hand, even in these conditions, you still observe partial DNA synthesis, then this is a strong indication that there is a structural obstacle in these myotube nuclei. And so, guess what's the result of this experiment? We isolated the nuclei from the myotubes, put them into the extract, and here are nuclei from quiescent myoblasts. Here are nuclei from myotubes. You see, by the end of the experiment, myotubes have replicated less than 50% of their DNA, while the myoblasts are basically at 100%. So, we were able to replicate incomplete DNA replication, even reproduce incomplete DNA replication, even in Xenopus extract, which, as I said, indicates that there is a structural obstacle in myotubes to the completion of DNA replication. Where do you go from here? Well, you'd like to know exactly what is that obstacle. So we thought, let's look for it. But before looking for it, we need to simplify our material. A nucleus is a very complex biological structure. We would like to have as simple a material as possible before we start looking for the obstacle. So let's try and get rid of whatever is not involved in determining complete DNA replication. Let's simplify these nuclei and get the simplest genetic material from myotubes that still shows incomplete DNA replication. That simplest material is the starting point to look for what causes incomplete DNA replication. So we thought of this. We can extract genetic material from the myotubes in various forms. It can be whole nuclei, as we did. It can be chromatin. It can be stripped chromatin, that is basically chromatin reduced to just nucleosomes. after eliminating all other proteins normally associated with DNA. And then, of course, we can extract crude DNA, pure DNA. All right, so let's put these different materials into the xenopus extract and see which one of them, compared with myoblasts, still shows incomplete DNA replication. So here's what we did. We put naked DNA, pure DNA, into the extract. Or we put nucleosomes, or we put chromatin. First experiment, pure DNA. As you would expect, there is no difference. After all, DNA is DNA. So DNA from proliferating myoblasts or terminally differentiated myotubes behaves just the same in the extract. Then we took the same nuclei as in the first experiment, but this time we subjected them to extraction with 0.5 molar sodium chloride, a very concentrated solution of sodium chloride. This strips the chromatin from of all its loosely or not even loosely bound proteins, except histones, which are very, very tightly bound to DNA. So basically, it reduces full chromatin to a necklace of histones and nucleosomes. And when we put this material from both myotubes and myoblasts into the extract, again, here are the myotubes with or without treatment with sodium chloride, showing that sodium chloride basically does not harm the nuclei. It strips the proteins away, but it doesn't affect replication. And here is chromatin, nucleosomes from myotubes with and without sodium chloride treatment. The two curves are superimposed. And again, you still see a major difference in DNA synthesis. So incomplete DNA replication is retained even in this very simplified material. So we know that with or without sodium chloride treatment, the incomplete DNA replication is conserved. But we still had the nucleosomes within the nuclear scaffold, even though I doubt they retain the original position in the nuclear scaffold. But anyway, still we have the nuclear scaffold around, that is the insoluble protein that make up the skeleton, let's say, of the nucleus. And we don't know whether the nuclear scaffold is involved or not in determining incomplete DNA replication. So what we need to do is to extract pure chromatin, full chromatin, containing all of its proteins, and test it in the extract, and then strip this chromatin and see whether sodium chloride stripped chromatin still shows incomplete DNA replication in the absence of the DNA scaffold. So we did this experiment, and here are the provisional results. We haven't completed these experiments, so this result, we do not regard this result as definitive, but it looks like even native chromatin still shows incomplete DNA replication in the xenopus extracts. Now, unfortunately, this is the last experiment I'm going to show you, and it's more than a year old. We haven't been able to complete our series of experiments because it's becoming more and more difficult. It's become already more and more difficult to find funds, money for basic science in Italy and elsewhere. And at this time, we are unable to continue our experiments, much to my consternation, of course. But we think we are close to our goal, and as soon as possible, we will start working on it again. Now, the conclusion of what I've told you. Terminal differentiation is indeed a distinct cell state. Some people have argued that terminal differentiation does not exist, that it's what we call terminal differentiation is basically a very deeply quiescent state that nonetheless can be reverted. Well, I think our data show that terminal differentiation is a state in itself, that in this state, the cells show properties that cannot be found in non-terminally differentiated cells. Cell cycle reactivation in terminally differentiated cells, plural, is now routine, which means we can reactivate them in many ways, and this is no longer a problem. What we need to know is what prevents terminally differentiated cells from fully complete their DNA replication. And so we are working, or we will be working on understanding the molecular nature of this problem, which we regard as the last problem before we can finally reactivate the cell cycle, the proliferation of terminally differentiating cells. And this is all. And of course, if you have questions, I would be more than happy to answer them.
Sorry, I have a question.
Please go ahead. Who are you?
I am Michela Orticello.
Okay, can you activate your camera, please, so that I can look at you?
Yes, of course.
Thank you, Michela. Go ahead.
Did you thought about you imagine some epigenetic mechanism that can be main responsible for this structural impairment of the DNA replication of some specific regions? Because it seems, as you've shown, something that is really structural, and probably some epigenetic mechanism can be involved.
Absolutely, it's a very serious possibility. You're right. It is very possible that the results I've shown you concerning chromatin can be explained by epigenetic modifications of the histones, not of DNA, because DNA, as you remember, behaves normally. DNA from myotubes. Now,
yes, at the level of the chromatin, so of course histone modification.
Exactly. But whatever epigenetic modification we are talking about must not be known yet, because all known epigenetic modifications are compatible with DNA replication. Of course, they are all represented in the cells we study all the time, and they all replicate their DNA completely. So if an epigenetic modification is responsible for what we observe, it must be an unknown one. Do you agree with that?
Yes. Yes. And this makes things much more challenging.
Much more what?
Much more challenging, because it's something that we still don't know. So it could be difficult also to decide what kind of approach to use.
Right, but did I give you the impression we are afraid of challenges?
Definitely not.
I'm kidding, of course.
And do you have an idea of which kind of epigenetic modification this could be, or you don't yet?
At this moment, I have no idea. If an epigenetic modification is involved, we should be able to identify it by using mass spectrometry. Other than that, I cannot tell you. I have no idea of what it might be.
Okay, thank you.
You're very welcome. Are there other questions or comments?
Can I ask, Marco, that you go back on a point which I thought was extremely interesting when you showed that the E1A reactivated nuclei keep their timing, whereas the cells that are inhibited in the CKI, in the kinase inhibitors, they lose the timing of replication.
Yes.
Comment more on that. Do you imagine a possible scenario for what makes the difference between those two types of reactivation? Because in one case, it's being more, you are taking a more global approach to actually perturbing all the kinase inhibitor system. In the other case, it's more targeted when you use E1A.
Okay, so I can tell you what we know about this. I don't have definitive answers, of course, or I would have told you. But what we know is that, okay, we ask the question, what's the basis of the difference between E1A reactivated and CKI depleted myotubes? What's the basic state of a myotube? Does it have, or does it have not, the ability to mount a DNA replication timing? That is the, oh, wait, sorry. Calling.
So I stop the microphone so it doesn't disturb you.
All right. So we asked again, what's the basic state of myotubes? Are they capable or not capable of mounting, of setting up a DNA replication timing? Because if they are not, then it means E1A is somehow reprogramming the myotubes into having a DNA replication timing. If they have it, then it means CDKI depletion eliminates a DNA replication timing that is already there, that is basically there in the myotubes even before they are reactivated. I'm simplifying, but that's the basic idea, all right? And the answer, the very neat answer is CDKI depletion suppresses DNA replication timing. If you double reactivate the myotubes with E1A plus CDKI in various combinations, E1A first, then CDKI depletion first, then you add E1A, the answer is always the same. CDKI are dominant. They suppress DNA replication timing. So, for reasons we do not understand at this time, suppressing the CDKIs in myotubes does reactivate the cell cycle, but also suppresses DNA replication timing. A DNA replication timing which would be there if they were reactivated by other means, such as E1A expression. We are doing similar experiments with fibroblasts to determine whether even there CDKI suppression impairs DNA replication timing. At this time, preliminary experiments suggest that it does, but not as profoundly as in myotubes. It is as if myotubes were a system in which these effects are magnified or are not hidden by other mechanisms that are absent in myotubes. So again, myotubes are a system in which you can ask questions that you wouldn't be able to address with other cell types. This is the best I can do to answer your question.
So that is extremely interesting in two respects, I find, because on the one hand, it says that the myotubes, even though they are not meant to replicate again, they carry with them the information that they had accumulated before during myogenic differentiation, and they sort of remember the timing with which the genome.
Exactly.
And then when you suppress the CDKs, you completely abolish that memory. So it's extremely interesting.
Okay, if you really want to know the whole truth as it appears now, we are not really erasing that memory. We are flattening it out. Because if you look carefully, you see that the myotubes, even if depleted of their CDKIs, remember what they were. The very little timing that it's left, very, very little, is the opposite of what you find in myoblasts or E1A reactivated myotubes. What is blue here is red here, and what is red here is blue here. So this is something we really don't understand, because it appears CDKI depletion does not only appear to flatten out the DNA replication timing, it also appears to invert its sign with minus instead of plus and plus instead of minus. And that really we have no explanation for.
Extremely interesting and important in terms of basic biology, as you were saying.
I'm fascinated by this.
Okay, so I think we should close the lecture now. Some of the students also have further lectures at 4 o'clock. And so, Marco, thank you very much. And as soon as we start a real program of seminars again, you should come back and give us some update on this work. It's very inspiring, very important.
It will be my pleasure.
Okay, thank you. And so to everybody else, we'll meet again virtually next Wednesday. And if you have questions for Marco, he gave you his references and contact points. So please, as usual, do not feel inhibited and ask questions, and go ahead with anything that you may want to have clarified. Okay. I'll see you on Wednesday then.
Goodbye, everybody.
Thank you so much. Goodbye.
Goodbye.
Grazie mille, arrivederci.
Thank you very much. Goodbye.
Thank you, goodbye.
Grazie ancora Marco, ciao. Allora, devo chiudere la registrazione, ecco. Bene.